good morning, everyone, or good afternoon, depending where you are in front of your screens. Super happy to introduce you to my four panelists for the first panel. Jeff Atkinson, Executive Director of Process Development and Analytical Science at Omega Therapeutics. Good morning to you. Then we have Angela Johnson, a Global Regulatory and Compliance Leader at Cytiva. My own colleague Lloyd Jeffs, our Senior Director of Biopharma Services at Precision Nanosystems /Cytiva, and Susie Jain, the Director for External Manufacturing at Cellular Therapeutics. Good morning, everyone. Let's kick off the first question. So generally what we want to speaking today is, we'll speak a bit about manufacturing of RNA-LNPs, regulatory challenges that are involved in this manufacturing pathway, a bit about commercial manufacturing. So basically, we will start slowly with process development and then make our way towards the regulatory phase together. So to kick this off, we all know that the industry, especially the RNA-LNP industry, has changed a lot during the last 2-3 years, especially due to the COVID pandemic that gave it kind of a spark. So I want to ask each of you, right, on your own take, and how has the regulatory and or commercial landscape evolved over the last year? What are your takes on that? And I would point out to Sujit first. So give me your take and then just make a round.
Sure. Thank you, Martin. Morning and again, afternoon, everyone, wherever you are. Thank you for joining us today. And then thank you to PNI for the opportunity. Martin, as you mentioned, the mRNA and LNP modality is not new in the industry, but again, there were not many products in the clinic or in the market as commercial until COVID mRNA vaccine came up, and now we're seeing that hockey stick growth for this modality. And then the demand for supply and manufacturing of these materials are still growing up. The capacity to make these materials didn't take too long to catch up. I think due to the fact the capital required to make or build the manufacturing plants for this modalities is not that high compared to viral vectors, for example. But I do feel maturity in terms of the knowledge and experience from the suppliers, CDMOs, even in the pharmaceutical product side, it's still not completely there yet, which obviously would take its own natural course of time. So I think it's for new companies, it's certainly pertinent to select the right CDMO for their needs, right supplier. I think some of the raw materials required to make these materials, we have seen due to COVID vaccine, right? The demand was so high, the supply and the capacity has caught up almost. A lot of the NTPs, enzymes, lipids, but some of the niche, I guess, excipients, if you will, niche based on your formulation, your needs. I think there's still some challenge in finding right supplier for those elements.
Yeah, that's perfect. Thanks for that. And it's great that you already touched a bit of like on the CDMO part, on the manufacturing, on sourcing material. That's great because we dig deeper into that in the coming hours. So maybe over to you, Jeff. What's your take on that? Because I think as an originator, you might have seen other challenges, other involvements, right, in the last couple of years.
So good morning and thanks, Martin. So yeah, I think that the capabilities have certainly increased within the industry. Several years ago, there weren't very many people that were working in the CDMO space, and it was harder to find that, as well as finding people from an innovator standpoint to work on these. It was a very, very new, very niche. So that's changed over the past few years in a really great way. So I think that's wonderful, and I think that the other part that is still evolving is learning the transition from the vaccines, which is where a lot of the headway was made, into the therapeutic areas, which were always there, but I think the real big splash came with the vaccines and really bringing it out to the public's knowledge.
Yeah, thanks for that, and I think that. Maybe Lloyd, you can come up next, right? So I know you're working with LNPs for only half of your life, right? So what's your take on it? How do you see the last few years compared to what you have seen probably the 20 years before?
Yeah, I'm really excited to see the blossoming of this technology. The fact that there's so many more people, so many more innovators getting into the technology, seeing its value, designing novel payload technologies, novel RNA and DNA technologies for therapeutic and vaccine applications, novel formulation development for indications that are quite wide-reaching, right? So, and this is a designed approach rather than discovery. I think it's a very systematic approach, and I think being part of Cytiva, I think I'm excited that we can be providing solutions to these innovators and the experts who are developing basically the next generation of therapeutics and vaccines for the world. And obviously, COVID-19, awful virus, and awful. A lot of people succumbed to the COVID-19 to the SARS-CoV-2 virus during the pandemic. But the ability to adapt and use this technology to protect billions of people from this virus, it was a real testament to the scalability, the manufacturability, and the overall applicability of this technology, right? So I think many more people now know of our friends, our relatives, even some of our elderly relatives actually know what a lipid nanoparticle is now. 20 years ago, over 20 years ago, when I started with LNPs, I was one of the pioneers in this space. It was a difficult conversation to explain what I actually did for a living, but now everybody knows. And I think so. We're, the team's growing, P&I, the overall expertise in the world's growing, and I'm really happy to see a lot of our clients and customers are becoming a lot more sophisticated about the details of this technology, the challenges, and certainly the opportunities. So I think it's a very exciting time for this field.
Yeah, I can absolutely agree with that. I only started 4.5 years ago with P&I, but also back then, it's even only 4.5 years ago, I had to explain my peers, my colleagues, my former colleagues, my parents, what I'm doing. But since then, everyone knows what I'm doing. They're like, oh yeah, you're working with LNPs. So that definitely has changed. And also professionalization of our clients makes my life also always easier, right? Because there's less for me to teach or more to focus on details rather than a broad introduction. That's great. And last but certainly not least, so Angela, maybe because the others also didn't touch that much on it, how do you see the regulatory space here has evolved? Because honestly, we had one product before and on the market. So also when I speak with the authorities here in Germany, when I have discussions with them on behalf of the clients, it's a bit, it was tough for them, right? Because it was kind of a new product for them. No one has touched it yet. So your take on how this has evolved, what have we achieved, maybe a bit what is missing, so we can introduce nicely that regulatory theme.
So happy to comment on that, and thanks, Martin. So I'm Angela Johnson. I've been in the industry a little more than 20 years, most of that working with novel delivery technologies, as well as advanced biologics, ranging from a variety of antibody products to cell, gene, and tissue engineering. I would really say, what's exciting is if there's any silver lining to some of the things that have happened in COVID, is that there were successes, internationally and at a national level. I look at things like Operation Warp Speed in the US and say, we really accelerated development in ways that shaved years off of what we've done in the past. What lessons can we take forward to that? There's still many challenges in the space, but we've opened up this conversation about platform technologies. LNPs are a great platform. FDA has formally put some efforts here behind platform designation and saying, let's make it easier. Can we reduce some of the toxicology requirements? Can we take some standard CMC approaches that really enabled you to get to those first in human and get through the development cycle faster, more efficiently, with more open information sharing, are going to be just a huge benefit for this industry and really enable innovators. So I think we're at a really exciting time. And I'll say, as someone who rode out the pandemic era at a small flagship venture company in that IND enabling space, I'd also say that we see a lot more emphasis on early industry agency collaboration, especially in the manufacturing space. FDA has created new meeting types, even expanded the Interact program, created the Type D. There's been expansions to the availability of those early innovation meetings in Europe and other regions as well. And I think this gives us a way to de-risk and validate the path forward we have for technologies like LNPs in a way that we simply couldn't do in the past.
Thanks for that. That's definitely interesting to hear. And then I can definitely recall that we also see that the willingness also from the authority side to learn and collaborate with the people working on this novel medicines is definitely there. It's growing. And I usually encourage also my clients to speak with them, right? To don't shy away with them, speak with them and ask for guidance early to not run into any bottlenecks. And I think it's great that you already dropped like kind of the buzzword platform technology, and that might be the next theme we want to go towards. So when we say platform technology, obviously, what do we need to really make LNPs a platform technology? So the first questions that I would have, and I would point to Lloyd first to give its take. What do you think are some key challenges in scaling an LNP formulation for GMP manufacturing? So what are key process parameters? Because obviously we need to understand those if you want to build a platform. So that would be the next question for you.
Yeah, so you got to look at what we're making, right? We're taking a nucleic acid, most commonly these days a messenger RNA, which is quite a large, it's a macromolecule, and using a recipe or a mixture of lipids to create a lipid nanoparticle. So there's at least 5 components in many of these lipid nanoparticles. And then there's the buffers as well that are required. And so it's very complicated and complex. And what we need to do is basically simplify the unit operations. So Precision Nanosystems has done that with our next-gen nanoassembler technology. So we've made it very easy for anyone, you don't have to be an expert in the field, to make high-quality nanoparticles. And that's great at small scale for in vitro screening and for more discovery work, for formulation development, to scale up all the way through to GMP and actually commercial manufacture of nanoparticles. So we have that part figured out. So the key process parameters for particle formation would be flow rate, would be the concentrations of the solutions and pHs, temperature that you're performing these steps at. There's many of those things that need to be controlled. Key process parameters we've seen for particle size have been the flow rate ratios. The particles are formed, as many of you know, by mixing a aqueous stream that contains nucleic acid with an organic, typically an ethanolic solution of lipids, and how they come together in a very controlled and precise way. That's the fundamental basis of our next-gen nanoassembler technology. So we figured that out for people, but you've also got to consider the downstream aspects. So off our instruments or systems, you get nanoparticles, but they're not ready to go into human beings. They're not ready to go into kind of an in vivo or even in vitro applications. What we need to do is actually remove ethanol. We need to concentrate down the samples while retaining the physical, chemical, and biological properties of the particle. So that's a challenge, and we actually have leveraged as part of the end-to-end solution, working with technology developed by our sister companies, and specifically within Cytiva for downstream processing, such as tangential flow filtration and sterile filtration. So there are kind of process considerations and key process parameters, both during tangential flow filtration and sterile filtration, and they need to be established for each formulation. So what we have seen, things like the choice of payload or the type of RNA payload may affect those. If you're using a buffer that will allow freezing and thawing of your LNP, you also have to consider the impact of that buffer on your downstream processing. So there's a number, and I don't want to kind of bore people with all the details, but I think sitting down with the overall process, whether you do that on your own or whether you ask P&I for some help, and we're happy to help, we can help you establish key and critical process parameters and actually generate the data to support the ranges for each of those process parameters. And last but not least is obviously we need to get this drug into a vial or a syringe for its finished product. So that's another consideration of the fill finish piece. And I don't want to ignore that piece. And I know there's people on this call that know a lot more about fill and finish than I do, and they can continue that on. But every step of the way, you could say the product is at risk, and we need to reduce that risk by establishing appropriate processing conditions, using appropriate equipment for each of these unit operations. So in order to accelerate the development of genomic medicines, specifically mRNA, LNP, you need to de-risk the manufacturing process before you can do the acceleration piece. And I see that a lot of the times where people have said, I'm going to take some shortcuts. I don't need to do this step or that step. I'm going to go from small scale straight to kind of at scale production. And it doesn't usually end very well. You know, shortcuts in our business, you know, you're basically going to come undone at some point. So having a systematic plan, understanding what's essential for development, generating data, batch data is tremendously important as well. So take a systematic approach rather than more of a like a Hail Mary where you're saying we're going to just do this in one go. Once you have that established, if you're doing other drug development programs, you can actually take that knowledge, you can take that experience, and that'll help you inform maybe for your second product. But first time through, be diligent, be really deliberate in what you're doing.
And that would enable you to do then basically also like a real platform technology, right? So if you have considered the right key process parameters, have understood them, then it's the chance for you to have a real platform that you can utilize.
Yeah, and I think
and the map, obviously, right? It's not like one fits all.
Yeah, and I talk to a lot of people in the and a lot of our clients, and I'm really impressed actually with that the knowledge and the experience that many of our clients have now that maybe didn't have 3 years ago or 4 years ago. So we got a lot of fast learners, and they've really adopted the technology and asking really good, tough questions, but really good questions as well. So we really appreciate that, and we welcome that. It's really going to drive this technology forward.
Perfect. No, that thanks for that, and I would give the others maybe an opportunity to maybe add on that. If not, I will put you on the spot here. So Lloyd already said you know more about fill and finish. So would someone be able to comment quickly about what the key considerations for fill and finish would be for LNPs? What are the challenges there? I don't know who would like to take that. Sujit, I see you want to take that. Thanks for that.
Yeah, I can certainly add a couple of things. I mean, I think I'm glad you connected the dots that, you know, how we can develop a platform technology. I think since I started on the mRNA LNP modality, I think one thing definitely seems unique to this modality, the formulation. I'm coming from biologics world, and then the formulation, many times we would have very, fixed candidates we would use, but now coming into LNP side, it's very construct dependent with your mRNA or other nucleic acid you are working with. So the lipid combinations could change drastically. I mean, certainly the helper lipids, PEG, they might stay, but the ionizable lipid, that could be very different molecule from one program to other program. But I definitely echo with what Lloyd said, to de-risk the manufacturing, the process conditions, the manufacturing equipments. You can definitely create a platform for those. I could touch quickly on the final piece, the DP fill finish side. The 0.2 micron filtration is the critical step I would consider for any drug product of biological nature, where we create that sterile environment, and this is your final step. So start thinking about those early on, the TFF definitely, but also 0.2 micron, right? You scale up based on pressure, delta P. So start doing those studies from your, scale up batches as well. I would just add, in addition to process condition and equipments, also keep in mind of your raw materials for the final drug product, right? Not just the lipids, but other excipients like your cryoprotectant, right? Make sure you use the right grade as soon as you hit, let's say, your GLP tox studies. You don't want to switch suppliers, switch the grade of those critical raw materials of your final drug product once you move into GMP, and then you have to show comparability from your GLP tox to GMP clinical trials and then commercial trials. So start planning for those raw materials, right? If your patient, for your indication patient population is higher, you have to keep that in mind, what vendor you're choosing for those excipients. I think those are certainly some key aspects. I think one should start thinking when they're planning for new products.
No, thanks for that. And actually, you already answered it. The follow-up question that I would have had for you, because the question is always, when I'm working on an R&D scale, right, when do I need to start to think about my GMP process? What do I need to design already kind of on the bench scale or a bit larger scale to be efficient and really have a GMP process like that work? But you touched on that already, so that's really great. You read my mind here. So we talked a lot about key process parameters now and what you need to develop. You talked about sizes, PDIs, and other things. So maybe Jeff, the next question is for you then. What do you think are the biggest analytical challenges, right? But because we can't find out the key process parameters and we can't detect them without a proper analytical frameworks. So could you talk a bit about what are the main analytical challenges around mRNA LNP characterizations and if there are differences between, let's say, an R&D product, the process development stage, and the GMP stage? So how does this evolve? So it's actually two questions: challenges and how does the analytical framework develop? That would be my two things here.
Sure. Yeah. So I guess kind of to answer the first part, right? That what's different between development and GMP? Well, in development, you're going to do a lot of characterization, right? You're going to try to figure out as much as you can, and then hopefully you compare back some of those assays as you move into more of your production, and especially when you get to your routine production, you would hope that you've figured it out by then. So some of the challenges from development and into scale up, both analytically and from a process standpoint, is the equipment has gotten much better over the years as far as scalability, but there's still a lot of, I'll call it art to what's going on here, right? You're mixing things together, and the analytical tools are still kind of catching up. There are things that we just don't know, that it's hard to figure out, right? So what does that secondary structure of the mRNA look like? Is it interacting with the lipids? How do we know how it's interacting? What are the analytical techniques that we can use for that? Potency is a real difficult assay to figure out. Whenever you're working with cell lines, right, and trying to figure out the potency of what's going on, it's difficult, right? It's not an absolute method. You're comparing against a reference standard that has its own set of issues. Ribogreen is kind of the industry standard, right? Everybody uses it, but nobody loves it, right? It's not a great assay. And you have to worry about interference. So anytime you deconstruct your LNP, right, you run the risk of creating an impurity that wasn't there in the state of the LNP. So how do you separate those out? How are those lipids packing? So we just don't have some of these methods worked out yet. So there's got to be a lot more looking at the methods. What instruments are out there? How can we use them in a different manner than what they're using? What are the new instruments that we need? And it does get a little bit back to what Suji was saying, like as far as going all the way back to your lipid manufacturing. Are you getting consistent lipids? How is your mRNA manufacturing going, right? What can you do back previous to starting the LNPs to eliminate some of those impurities or problems that could be carried forward into your LNP? So you have to start with your methods there, and because this is basically you're encapsulating it, right? So how do you have to start out with analyzing all of your different components and then manipulating those methods and adapting them to the whole LNP, and then trying to separate it back out again without disrupting anything. So those are some real tough challenges to get behind. And it's just a lot of work up front. Once you figure those out and look at, how your integrity may be affecting your potency, how those lipids may be interacting. So there's a lot of development work up front. The biggest challenge is, what do you do? Like, how much is enough to feel like you're ready to go to the clinic, and how much is enough to go to the agency with, right? So those are real challenges to figure out. But eventually you want to get to a point where you can say, we've done all this characterization, we understand what's going on. Now let's go back and say, these are things that we no longer need to be tracking, or we track them on a less frequent basis.
That absolutely makes sense, and it's good that leads us to you, Angela. So I would like to commend you a bit on that. So let's say we've done all this great work, right? We have fixed our analytics, we have fixed the manufacturing process. So up next would be we need to do the IND filing, or we need to file for clinical trials, right? So how do you feel? How standardized is that already, right? What we can do here, what we need to evolve, where should we really be focusing on when we work towards the regulatory phase? So that might be what I need from you at this point to kind of wrap this together.
So that's a really important question right now. And I think it really does vary here. There's a difference in strategy between perhaps vaccine development, and if we're going into something like gene therapy, gene editing, immuno-oncology, in terms of what these programs look like, whether they're going into a traditional phase 1 or something more phase 1, 2, where there's a higher level of CMC readiness that's an expectation. So I would say very much looking at the requirements within your area and taking a phase appropriate approach. And to touch on platform technology pieces, some things we understand well, some of the formulations that do exist out there that are well studied. What's less understood is targeting other organs. I'll say that could be very different immunogenicity profiles, biodistribution, toxicities that aren't understood. And when we're going into a really novel space, I'll say it's important to plan for those things, have those early engagements with the agency on when it'll be done. Is that before an initial IND? Is there pieces that can be done in parallel or leveraged from a similar formulation? How do we put together that best data package? I think is really a lot of the work that we have to do in the industry right now to find what can we leverage across these platforms as they mature and as they expand, and what really is unique to each and every therapeutic application of LNPs that we're going to be expected to do each and every time for new products. And I think there's a lot of movement in that area in particular. The only other comment I would make is being very strategic. We talked about process changes during that IND enabling period or a CTA enabling period, is saying, when can I go into my animal models, my non-human primates or large animal models? What's the earliest I can do it, and what's the risk? Could I go earlier if I do some comparability work? Is it minor changes in formulation? Does this need to have a conversation with an agency to validate a perhaps streamlined development approach? Is a conversation that we have quite a lot. Yeah, always a risk benefit there.
Yeah, thanks for that. And I think we had now a nice first part of the session. So I have a tough challenge for you now. So each of you commented on your own thing. On your opinion, each of you just tell me once, what's the biggest challenge in your area, and a tip for, or the top tip for people. Really only one sentence each. So I don't know who wants to start. If not, Sujit, can you start, or Lloyd? Yeah, Lloyd, go ahead.
Yeah, I can start. We need to do this quickly. I think time, and obviously time and money, are inexplicably linked. And for drug developers, they need seamless end-to-end solution, minimizing time. And I think what I've seen, I worked in the drug development space for about 17 years before joining P&I around 5 years ago. That was always the biggest pressure. We need to get here by this point. We need to have an IND submitted by this point. We have this much cash runway. We need to make the best use of our time and our money. And
what's your tip here? Where to focus to make that happen? It's a pretty broad here, but still, where to focus to make that happen?
The tip is that, oftentimes you might be working with 8 or 9 different subcontractors to actually bring this all together. That's a lot of management. If you can reduce that number to 2 or 3, I think that's going to help you. That's going to help you move your timeline and your program forward.
Perfect. Thank you for that. I don't know, Sujit, you want to go next?
Yeah, certainly. I can just build on what Lloyd said. But this is still complex field, right? With this modality, I think my tip first, I would just build on that. Start early. As Lloyd said, there's so many suppliers. There's so many elements you have to plan and think about. So the tip would be to start early. Start early on thinking of scale up, thinking of your GMP supply for your raw materials, critical raw materials, cell banks, lipids, all of that. And if you can find a vendor, a CDMO, right, end to end, you can minimize those number of suppliers, that's certainly ideal. I think the challenge is basically that right now it's just, as I mentioned earlier, on the maturity. So there's just so many players that you have to figure out which ones are the best for you.
Awesome. So Jeff, your main challenge and your tip how to overcome it.
Yeah, so I'll try not to repeat, because I think we're all going to basically say the same thing. So definitely time and your money runway are big, and looking at that supply chain. I'm gonna go in a little bit different direction. That is, it's still do it early, but looking at stability and just the practicality. I mean, the industry standard right now is -70 for most of these mRNAs, LNPs, and it's difficult, right? From a commercial standpoint, is that really where we want to be? So understanding that stability early, because that can really hurt you. And then, so I guess that's the tip, right? Do lots of stability as early as possible, and try to figure that out. And long term, hopefully we can come up with something better. I know there's a lot of people working on lyophilization. So
yes, they do, and I think that's also a good tip too, because I see that too, that is a bit underestimated, right? Everyone figures out the biological kind of the manufacturing pathway, but then when it's time for the IND filing, they figure out, oh, we need to look at stability, and stability is also a thing that needs time, right? If you need 3 months of stability, you need 3 months. If you need 6, right? You cannot shortcut here, because time is time. Yeah, totally understood. That's great. So Angela, you're the last one for that round.
So I'll answer it from just a little of a different angle too, and say we've had this concept in regulatory that you may have heard of called sameness. 10, 15 years ago, we were talking about it for orphan products. We talked about it for cell therapies within the last decade, and we saw a fairly recent gene therapy guidance come out in the last few years on what is really the same mean in a platform. And I think that's where a lot of the ambiguity lies. We know we can save a lot of testing and developmental work if we identify that this is the same or similar to other formulations, and what those regulatory expectations are, both in process development, IND enabling, and throughout the clinical development cycle. Right now, I think, though, that's really unclear. There's no formal standardized guidance out there for it. I think that's maturing. And right now, while we're in that maturation phase, is having those early conversations so you identify what is the same and aren't investing, I'll say, in costly and time-consuming efforts that are duplicative or not necessary for a program, and hitting that right and somewhat subjective bar that may vary between regions.
Oh, thanks a lot for that. And I think that kind of wraps up the first part of our session, talking about a bit process development, how to get to that IND filing and really get into the clinic. So let's say we made it, right? We're in the clinic now, and now it's really time, or just before we inject our first patient, now we need to manufacture our drug. So I think I have the first question. Let's talk about single use a bit, right? So we know that especially also a lot of biopharma, traditional biopharma manufacturing for mAbs, for example, they use single use technologies. So how do you guys see the use of single guise technology also in the LNP field? What benefits does it have to use a single use technology? And I think maybe Sujit, maybe you can start with your thoughts on that.
Yeah, definitely. And certainly I have seen personally moving from stainless steel to single use for biologics too. So single use definitely has a lot of benefits, right? And those apply for mRNA LNP as well. If you're trying to achieve speed to clinic, or even speed to market, right? Single use is your friend. Certainly, we had seen some challenges in the supply chain, but I think long term, for this is going to stay. And from mRNA in the IVT, production in a single use bags, certainly you have the tubings and the storage and holding bags, but even for LNP and then TFF, you have those single use membranes, pre-packed columns. I think single use provides a lot of flexibility. You don't have to worry about cleaning, right? The timeline to reach scale up and different scale of batches, it's very quick. There's no cross contamination if you're producing multiple batches. So even at a CDMO facility, you don't have to worry too much between client and client's product contamination if you use single use. And then there's no downtime. You can switch over very quickly. And so it's the same benefits for mAb. I think those apply similarly to LNP as well.
Yeah, that's great. Thanks for that. And maybe I want to have a follow-up question, because yeah, I agree. Single use is the right move for LNP. It's also looking at how costly these raw materials are, how costly RNAs and the lipids. You want to also de-risk that whole process. And also looking at how many studies will come in the clinic, you might need to switch quickly, right, between similar projects or different projects and really manufacture them quickly. But do you think there are still challenges with single use? Because what I hear a lot out there is using ethanol with bioprocess specs. I see Lloyd nodding here. So if you could comment a bit on that, I would appreciate that.
Yeah, so obviously material compatibility of any single-use component is important and needs to be considered. You need to do an assessment. I'd say another consideration is with single-use technology, I think Sujit spoke to this a little bit, is understanding the lead time for each single-use component that you have, and then also understanding the shelf life of each single-use component. Because if you keep a large inventory, you do run the risk of actually having your single-use fluid paths or have you expiring before you can use them. For an R&D setting, probably less critical, but it's absolutely critical as you get into kind of larger scale batches that support IND enabling studies, GLP tox studies, and especially for GMP. So I think managing that from a supply chain perspective is critical. And I know a lot of small companies, supply chain's done by the scientists, the developers. They don't have, a large supply chain group. So that's something that I didn't, in my training as a scientist, I didn't really consider that. When I joined the industry, that became a massive consideration. So, it's important to keep that in mind when you're planning out your studies. And again, starting early, getting a good plan, understanding what you're going to need now, what you're going to need in 3 months, what you're going to need in 6 months' time. Don't forget that component, because it's not like Amazon, right? You can't just kind of pick up the phone and get a next day delivery for a lot of these items. They are kind of specialty items, and there's quite a large demand still for these items as well. I know some of the inventory levels are much higher than they were during COVID-19, which is good, but there's still long lead time items for some of these single-use materials, so keep that in mind.
Thanks for that. And I don't know, I see a lot of nodding here, Jeff. Do you want to add something? If not,
I can add a couple things. As somebody that was working in the therapeutic area during the pandemic, right, that single use became a real issue from a supply standpoint, because everything had to go towards the vaccines first. So it was a challenge, and look, I think that they have a fantastic place, and they're really, single use is definitely a great way to go. It's just a consideration, right? To choose your supplier, as Lloyd was saying, like, be on top of it, know when you need more of that material, and have a safety stock. You may, and unfortunately, it may mean that you lose some of that to expiry, which is not great. But the role is there. And if your quantities are large enough, maybe there's a role for a combination between single use and more permanent equipment that should be considered.
Awesome. And Angela, maybe I have a follow-up question for you here. So when you say, so what's your take on single use, right? Because we talked a lot of the benefits were really manufacturing related right now.
Yeah.
But can you talk a bit about also from a regulatory side, if single use makes sense and what it might ease or what it might complicate to use such a technology?
So I'll take that from two angles, and coming from a similar perspective as Jeff, as someone who spent a lot of time in clinical development, both in ClinOps and regulatory, I'll say that, there's some obvious things you'll need to do there. There's USPA requirements, extractables and leachables, materials considerations. I think we hit this a lot in the pandemic where supply necessitated materials changes, and then there's a bit of a scurry to say, what else do we need to do because of a necessary shortage related materials change? And that's something that does happen. But with that, I want to sort of turn the focus to the clinical side here and say, in a lot of these areas we're developing in, especially the exciting areas that are opportunities for LNPs, to me, it's very competitive from a clinical standpoint. Identifying patients may be hard. Sometimes there's hundreds or thousands of competing trials. So I see one of the biggest risks in clinical development is identifying those patients, often at a very high cost, through CROs, partners, university research sites, and then not having product available or having loss of products out of spec, and saying, I have a patient, I've incurred the cost, but we can't actually treat that patient, or either they're lost because they can't receive a follow-up treatment somewhere down the road. But those can both complicate the data as well as extend trials and even lead to failed trials in the worst case. So I think single use has a lot of benefits here. We prevent some of that loss from possible contamination, batch to batch carryover issues, which are an issue at CMOs working, I'll say, in multiple products. And I would say single use in general provides a lot of opportunity here to minimize the risk that we're going to lose clinical product if the supply is done correctly. And you have to balance, I'll say, some of the manufacturing considerations versus, I'll say, both the time and the direct cost of losing patients in your trial.
Yeah, that absolutely makes sense. And I think maybe it's great that you kind of touched on that already. You said the risk of losing parts or entire batches. So maybe we can talk a few minutes about what can we do to ensure that the manufacturing process is that optimized, that we limit batch to batch inconsistency, and that we make sure batches always succeed. And not really sure, but I think maybe Jeff, you can start this time. So how can we ensure that we really get the batch produced and get it into the patient time after time?
Yeah, so I mean, that is going back to, your process, right? How consistent can you make it? Do you know what your parameters are early on, identifying those, and then choosing the right analytical methods? Analytical always has to lead the process, right? You've got to know what you're making, so you have to figure out something there, and that way you can characterize it and make the same. You're choosing a good partner in your CDMO, right? Somebody that has the right experience, that has done this before. There are a lot of people that have gotten into it and are trying to build the capabilities, but there's still not a lot that have a lot of expertise, right? So it's be careful in your selection of a CDMO, and as much as you can build that capability in-house so you understand what they're doing, and that you can transfer a good process to them. So I think that's really key, is to try to understand it best you can and move on, unless you can find a partner that has that expertise. And sometimes you have to rely on those partners with the expertise, right? Like that's part of the business model for a lot of companies. They have to go there first. And that's perfectly acceptable. Choose well, like look for a really good partner.
That makes sense. Thanks for that. I don't know. Does anyone of the others want to comment on that? If not, I would switch gears a bit here. So I think the most things we spoke about, and that's what, when we think about LNPs, a lot of people think about the vaccines, think about that large scale manufacturing, right? But what I see, and that's probably a trend that you might see too, the next studies that will come up are a step more personalized, right? Smaller batches, and that will evolve. So I guess looking out, 2, 3, 4, 5 years in front, we will have to manufacture maybe even personalized patient individual batches. So maybe that's for you, Suchit. How do you feel the landscape evolves? Plus, what would be the challenges from moving from a large scale process to a small scale gene process? Are we ready for it? What do we need to be ready for it? Yeah.
No, that's definitely a good question. I think we're all thinking about that. I think the whole industry is moving towards, you can see, for personalized. And maybe I think there is some benefit we're having for this modality because we have the CAR T experience. So I think there is some experience from manufacturing and compliance, how to make this personalized, manufacturing batches. I think one good thing with the mRNA or nucleic acid and LNP, right? I think it's from scale perspective, as Lloyd mentioned, there are tools available already where you can seemingly see the scale up performance. So you could still have a small scale equipment, which performs very closely or similarly to your scale down batches. So I think the tools we have, those certainly make this a reality. And then our prior experience with CAR T and other gene therapy modalities, I think we have a precedent on how the quality and manufacturing compliance need to be in place to make such personalized batches.
That's great. So I don't know, Lloyd, maybe you can comment on, do you think we have the manufacturing equipment ready for that? Or is that something we still need to amend and adopt to really produce, let's say, a 10 milligram batch?
I think there's work being done on that. There's been certainly a lot of development. I think what we are seeing now more than ever is for this, mRNA LNP technology. I think historically people have taken existing technologies and adapted them. Whereas now there's more of a deliberate effort to say, okay, this is the requirements for an LNP technology. This is the equipment we need. These are the types of filters we need, or cartridges or cassettes for tangential flow filtration. I think there's a lot more work being done in that area rather than just taking stuff off the shelf that's been developed for other applications. So I'm very excited about that. I think that would apply obviously to personalized medicine, because at the end of the day, you need to make batches very quickly at small scale. Everybody is concentrating on scale up, like how can we make billions and billions of doses of mRNA LNP vaccine? And you need to be able to scale a process both ways, especially in personalized medicine. You need to scale it down without losing any of those properties. So scale down, I think, is going to be the challenge. And when you scale down, you've got less room for error, right? If you're making a large batch, you got to think about hold-up losses and what have you. They're going to become insignificant at like hundreds of liters of scale. When you scale down, every drop counts. So to do that in a way that's more automated, that takes away the human factor, and to do that very quickly and simply, I think is going to be the kind of the driving toward that solution. Where and if you can do that at the bedside or in a hospital clinic, that's ideal, right? You can't, you want to basically set up the equipment to make this personalized medicine very close to where the patient actually is. You don't have to ship it halfway across the world or halfway across the continent to get there, right? So you want to eliminate things like basically the having to store things for an extended period of time, having it on demand, and actually having kind of a regulatory framework to allow that to happen, right? So I think there's certainly some challenges there, but I see that in the future, and I think that's coming quite soon, actually, given the breakthroughs that we're seeing.
No, that's great feedback, and I think that already brings me to and Jeff, Angela, you need to collaborate a bit now on that piece. So when we look at release testing, traditional release testing for these bigger batches, right? There's a lot of analytics involved, and obviously for personalized medicine, we can't do that, right? We can't have a clinical hospital to do 30, 35 analytical methods, right? There needs to be some standard methods that they can do. So maybe your take, Jeff, first on that. How can we develop the process in a way that simple analytics would allow to do, and what analytics do you feel would that be?
Yeah, I mean that's
probably a tough one.
Yeah, that's a really tough question. So yeah, I think that there are some things that we can do, but it would come more back to what equipment are we using to make it? How can we make that consistent? What online parameters can we be monitoring? So that it's more of a parametric release, right? Like when it comes off the end, maybe we only have to do 2 or 3 things, right? Like just quick checks, verifications, and then it's ready to go to the patient. So I think it's got to be completely integrated with the manufacturing, if you want to call it that, right? It's on-site manufacturing of the product, and have all of that analytics on board. So that in my opinion, that's the only way you get there. You can't train up all of these people to be doing our normal array of tests on LNPs. It's just too many, and they're too complicated.
All right, thanks for that. And Angela, then maybe as a follow-up from your side, do you feel how the regulatory framework is set up right now, is it ready for personalized medicines, or is there really challenges still awaiting when we want to put an LNP in a personalized medicine framework? We know we have it for CAR T cells, right?
Yeah. So I think that's a loaded question in many ways, but I will say there's still many challenges remaining, but I think there's a general recognition that ultimately decentralizing that manufacturing is going to be key, especially with products with unique personalized characteristics, or I'll say, unique transport, storage considerations, making them globally accessible. This is really going to be the key to accessibility for many patient populations in the long run. Conceptually, I often think of testing. It's a way to detect process irregularities or errors that may have occurred. So the more certainty regulators get in the process itself, as that's established, as we have good data, as we deploy maybe modern CMC, non-paper-based sensors and analytics, we can really get ahead of those type of process errors and increase our regulators' certainty, that these products are at the end of the day safe and that they are working as intended. I'll say, and somewhat reduce those testing requirements in personalized medicine, which is something that's, I'll say, across the board an issue in personalized medicine. But are we there yet with LNPs? No, but I think as an industry, that's the conversation to have. And one of the good formats, especially when we hit on the unique equipment development. And, I think this is something unique about Cytiva and Precision Nanosystems, that we're able to engage with the regulatory agencies at tech teams or at working groups and really have these early conversations around what kind of equipment changes or innovations, might we work on in the future? What kind of technologies are they willing to give us early feedback on? That can be really beneficial. And I'll say those early, even before product development, tech team style meetings can give you great insights into regulators' thinking years before the guidance, or I'll say some of that binding standardization comes out.
That's awesome. No, great. So that would, I think it's a logical step, right? Because RNA medicines or nucleic acid medicines are made for curing very specific diseases, very specific genetic defects in patients. So I think for me, it's the next logical step to treat people on that molecular level, really what they're suffering from, and not only in a broader spectrum. So it would be amazing to really implement that. And I think that brings me a bit to, we only have a few minutes left, but I want to talk quickly about that. So we talked about cell therapy, about vaccines, about maybe other diseases. So quick answer, maybe Lloyd, you can start here. So what do you think are the differences or the challenges unlocking these other targets? Maybe having an LNP for cell therapy, having an LNP for curing a liver disease, having an LNP for a lung disease. What would be the challenges here?
Yeah, I think you have to ask, that's a great question. I think fundamentally, why do we even have LNP? Why don't we just inject mRNA into cells directly or into the bloodstream? And it's because you need a delivery solution. And for those different applications, you're going to need novel delivery solutions. So again, one size does not fit all. And I think it's super exciting because we've got a lot more people now thinking about novel LNP-based and nanoparticle-based delivery systems than ever before, right? How do we, original formulations were very good after IV administration at going to the liver. They were optimized and developed to do that. For vaccines, obviously it's intramuscular injection. Is it a big challenge for delivery? Not really, as long as they can get into the cell type and release their contents into the cytoplasm intact. But again, looking at other disease sites and diseased organs and tissues, that novel delivery is critical, and it might be the choice of your ionizable lipid. But there may be targeting moieties you might want to use. You might want to modulate the size of your particle, making it bigger or smaller. There's so many things you can do. Most of that is in research right now, but I see that kind of a lot of those advances evolving from research and transitioning into more development and hopefully into the clinic soon. I think you're seeing a ton. We see this at Precision Nanosystems. We're seeing a ton of really neat ideas that are progressing and being advanced. So, I'm looking forward to this innovation from drug developers in the next 12 to 18 months. You're going to see some major breakthroughs. I'm very excited about this.
Awesome, thanks for that. I don't know if anyone wants to comment on that further. Yeah, I see Sujit, go ahead.
Yeah, certainly the novel, delivery, that's right now a missing piece, right? Delivery has been the key to solving, gene therapy for the last 10 years or so. Certainly we started with viral vectors, now mRNA, LNP, but we still have to achieve that targeted specificity. So the novel solutions would certainly come in play But I do see the challenge in terms of even the supply and manufacturing. What we are facing with this novel modalities, right? We're looking into not just small molecules, now peptides, even antibodies for targeting. So how do we get those, in our supply chain? How do we make that material, but also together with the LNPs? So that's, I think, I don't know if anybody or a lot of people have tried that yet in the clinical GMP grade environment. So that would be a challenge from my perspective. How do we make this, in the GMP environment for patients, for clinic and commercial, let alone the supply of the raw materials needed for these. And I think the other side I do want to add, Salugen, we are working on not just mRNA, but other modalities used for gene delivery, like DNA, right? There's certainly DNA vaccines, but DNA for therapy, I think there's still some challenges to deliver DNA, which is not similar to mRNA. You need active delivery. So that's where the novel solutions would come in. But there's still more room, definitely.
Awesome, thanks for that. And we're almost at the top of the hour. So I want to ask each of you the same closing questions, and you only will have slightly below a minute to answer each of you. So how do you feel the field evolving the next 5 to 10 years? To make it maybe a quicker answer, what development are you most excited about to see in the next 5 to 10 years when it comes to mRNA LNP? And this time I want to start with you, Angela.
I'll be short and sweet, and I'll say a better way of performing gene therapies that has a better durability, perhaps retreatment, really allows us to do this better than viral vectors have in the past. We've seen some amazing results, and I think this has the potential to really come to the clinic in the next decade as a truly changing of the healthcare field kind of treatments.
Awesome. I just go around my screen now. Jeff, you're up next. What are you excited about next 5 to 10 years?
So I'll kind of echo what Lloyd and Sujit said, right? Like that targeting. We need better targeting, non-hepatic targeting. So that's where a lot of work is, and we need a lot of help from biologists to get the right targeting agents to put on those LNPs. And I'm really excited. I know there's a lot of work going in that area. I'm excited to see what comes out.
Awesome. Lloyd, your take. What are you excited about?
All of the above that's been previously mentioned. If I had to pick one, I'm really excited about personalized medicine.
Yeah, me as well. I think that's also where I see best fit for the technology and huge game changer. Thanks, Sujit. You answered my first question, so you have the honor to also really answer the last one. So what are you excited about for the next 5 to 10 years?
I'm actually going to add maybe on the other side, which Lloyd and Jeff earlier mentioned. I think the challenge of, why do we need LNP, a lipid nanoparticle, right? Can we just dose the payload as is, right? So a stable, I think, payload like mRNA is not stable. So something which is more stable, and it could be like a conjugate connected to the mRNA, and certainly the other circular and other kinds of RNAs are coming into picture, which are still in R&D. But I see that probably would be next wave of innovation, that with the payload itself, how do we modify them to be more stable? But also if we could just dose them directly.
Awesome. So we made it perfectly. It's top of the hour. Thanks again for all of you for being here, taking the time, answering my sometimes very tricky questions. I hope the audience enjoyed this panel.